All right. Me. All right, thank you for coming back. We are going to our second part, and we have with us Jacob. Jacob is an analytical chemist. He's based at analytical laboratories in Hamilton, and that's one of New Zealand's newest and most innovative high-throughput analytical testing labs. He finished his PhD at Waikato University, studying the catalytic properties of metal nanoparticles before moving to analytica in 2013. Since then, he has worked on targeted and untargeted mass spectral methods for measuring the composition and authenticity of honey and dairy products by mass spectrometry, as well as an array of other matrices. And today we're going to talk about mass spectrometry in a commercial environment.
Let's get that going. That'll be important. Thank you very much. How long do you want me to talk for again? Forever?
Yeah, you have pretty much 4 hours.
Okay. Well, thanks for inviting me, everyone. It's a pleasure to be here today and talk to a captive audience about mass spectrometry, because not everyone necessarily enjoys hearing about it. My partner certainly always glaze over whenever I tell her about peaks and calibration curves and things. So to talk to you guys about this is great. So I jumped at the opportunity to come here and do it. But in retrospect, afterwards I thought, oh shit, actually this is going to be quite a difficult talk, because a lot of you guys are from the academic space. We're used to reading journal articles where everything is laid out in exquisite detail about exactly how experiments are done and what the methods are and what mass transitions are used and how the chromatography works and all that sort of thing. Conversely, since I'm from a commercial testing lab, we actually like to do the opposite. We like to keep everything a secret, and we don't like to tell anyone about how anything works. The reason for that being that a lot of the stuff, a lot of the processes and methods and things that we've developed have been done in-house, and we think they confer some advantages over what competitors or other laboratories are doing. So there is, of course, motivation for us to be quite secretive about it. So I thought, oh, I could tell them about that. Oh, no, I can't tell them about that. I could tell them about this one. No, I can't do that either. Oh, shit. Okay. Actually, I won't be able to tell them about a lot of the stuff. So I hope you will excuse me that a lot of the stuff that I will talk about is kind of really around the periphery or the edges of mass spectrometry, and there isn't quite so much detail as some of the previous speakers have given you. So, Analytical Laboratories, we are based in Hamilton in New Zealand. We started in 2012 with 2 or 3 people in a space rented from Ag Research. And since then, until today, we've expanded. We have about 140 staff now. Hamilton is still our main location, but we have a handful of other satellite labs around the country that do non-mass spec things like asbestos testing. Water testing, etc. So in terms of what we do at our main lab, a couple of different areas. First one, drugs. And I don't mean we do sort of sensory organoleptic testing of drugs. We do things, we test housing swabs for presence or contamination of amphetamines, like methamphetamine. And we also do a little bit in the cannabis space as well, testing various products for cannabinoids and terpenes that give cannabis and hemp its unique profiles of smell and taste. We do quite a lot in the dairy space as well. So we do testing for a lot of dairy companies for a lot of different things, for residues that shouldn't be in different dairy products, or that dishonest people are putting in dairy products, and as well as for things that that give certain companies or certain dairy products an edge over others. The other 2 areas that we operate in are honey testing. So we do a lot of testing on different honey types, mostly Manuka honey, which is a very valuable industry in New Zealand, about $400 million a year worth. And of course, there is a lot of interesting things to test for there. And the fourth kind of space that we work in is the environmental space. So we're testing soils and wastewaters and kind of industrial waste for things like heavy metals, organic contaminants, petroleum byproducts, etc. So we are a high throughput routine testing lab. And what does that mean? What does that mean? That means that we are trying to provide our clients reliable information, means as accurate as possible, as reproducible as possible, and in a rapid fashion. So to some people, rapid means you can do it in a month, or you can do it in a week, or maybe you'd be lucky if you can get all your MB research done in 2 years instead of 4. To us, high throughput and rapid means that once a sample comes in the door, we give someone a result for it within 24 hours. So if it comes in 9 a.m. one day, the next day, that person has the result on their, not desk, but on their metaphorical desk, in their email. So we call this high throughput routine testing, and there is a lot of tools that we use to do it. Now, mass spec, although we would like to think it is the be all and end all of detection mechanisms, it is just one of the tools that we use, but it is the one that I will focus on today. So the general workflow is something like this. So someone will send us a sample of a food, for example, a honey like this, and they'll write down what they want it tested for. So we have a suite of predefined tests that have been set up and very, very detailedly validated to document the performance and how they're done. Sometimes people ask us for particular tests. Sometimes people don't even know what they want. They just ask for it to be tested. Okay, sure. We'll call and then dig into that one a bit deeper. The next step is that we prepare the samples and we run them through some sort of a science machine, like a mass spectrometer or a chromatograph of some description. I tried to find clip art of a mass spectrometer, but unsurprisingly, there's not a lot of good mass spec clip art on Google, but I was quite surprised to find there is something that pretty closely resembles an HPL, so we'll go with that as a sort of surrogate. So we prepare the samples, we run them through an HPLC or a mass spec or something with a whole bunch of different checks and quality controls. And the final result is that we output some sort of information like a report. And that's basically the end of the process as far as the testing is concerned. Of course, at some point, someone has to pay us some money for what we have done. Some people think that high throughput routine testing, you would be rolling in money like a Scrooge McDuck type of figure, but it is a fairly lean operation. It's not necessarily making us all millionaires. So that's what it is. But there's a couple of things that high throughput routine testing with mass spectrometry isn't. And these things can be quite surprising, especially to people who come from academia. Maybe not surprising to people who've been in this area though. So the first thing that it isn't is it is not about taking your results and making them publicly available to everyone in the form of some sort of beautiful journal article or presentation. It's absolutely not about that. For the reasons that I mentioned at the start, a lot of this information is proprietary. We don't want to spread it. It's what makes us money and pays our rent. The second thing that it is not is a charity organization. So some people think that they shouldn't have to pay anything for testing, that they've done all the hard work collecting their particular samples, their food, their whatever it is, and we should do the testing for free. It's outrageous that we charge $50 to test a sample for something and have to pay for all our rent and millions of dollars of equipment and lawyers' fees and all that sort of thing. So it is definitely not a charity organisation. We're trying to make, we're trying to provide clients good, valuable information, and in the process, we are also trying to make money as well. So let's talk about some characteristics of routine testing. When we say rapid, like I said, that usually means we're sending results out 24 to 48 hours after we've received the sample. Anything longer than that is getting a bit slow for some people. There's a lot of very impatient people in the food and industrial sectors in New Zealand. People can't wait a week or 2 weeks or 4 weeks to get results. They have to hold these samples and wait until they've got our result before they can do anything with them. To them, that's lost money, and to us, it's not a good situation to be in. So for this rapid testing, depending on what the test is, we're looking at sort of doing 100 to 1,000 samples per day for a particular type of test. Usually that uses one or two different pieces of equipment or different instruments, different mass specs, and it usually uses a fairly small number of people. So a small team can knock all that out and get all those results out the next day if everything goes well. Now, the other component of routine testing, of course, like I said, is making sure that everything is reliable. So if your data is not reliable, then no one is going to come to you. You're not going to be able to make any money. You're not going to be able to buy any cool mass specs to play around with. So there's a lot of quality control that's involved in this testing. There's validation of methods following strict procedures. There's accreditations to make sure the laboratory is doing what it says it is, and all the equipment is fit for purpose, and so on and so forth. And there's lots and lots of controls that go along with every single batch of samples. There's duplicates, there's spikes, there's positive and negative controls, there's all sorts of stuff. So there's a lot of rules around how the stuff has to be done. And when I first started working there, I thought, oh, there's lots of rules. This will be a piece of cake. This will be like running a train. Like, it's stuck on the rails. You shovel coal into the furnace, away she goes. Not actually like that. There's, of course, as always the case, a lot of problems that come up, and a lot of challenges that we need to face. So in terms of how you get throughput with something like this, there are three kind of key technologies really that we utilize to do this. So if you're doing your masters, for example, you might be used to running all your samples in vials, having to wash the vials afterwards and recycle them because your supervisor won't pay for a new box of them or something like that. We don't do any of that. Everything goes in 96 well plates. It's been mentioned before, but 96 well plates are your friend, because every time stuff goes in a plate, it's more vials that you don't have to label and less mistakes that you can potentially make as a consequence of it. So you either fear 96 well plates and you lose, or you make friends with them. Now, the second key piece of technology is liquid handling solutions. And what that is basically a fancy way of saying that you replace an army of 50 technicians who would be laboriously pipetting stuff all day and getting RSI in their wrists with a single robot that costs you between $100,000 and $400,000. But a robot like this, you can shovel samples into all day long, and it'll move them to where they need to go. It'll do dilutions, it'll add derivatizing agents, it'll do cleanups. It will never complain, it will never take sick days, and it will never make you annoyed. Actually, that last one's not quite so bad. And they can be annoying sometimes. Now, the final one is what I'm referring to here is redundancy. There's probably a better word for it. But what it really means is that instead of having sort of one of all of the different types of mass specs, like you might have at a university, we have, say, 10 or 20 of a single type of mass spec. So we don't have one moldy, one FTICR, one Orbitrap, one triple quad, one this, one that, da, 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 da. We have like 5 or 10 of the exact same technology. And what that means is that you can set up a method using one piece of equipment, and when it falls over, when it goes down and something breaks and you have to fix it, there is no downtime. You can just move it straight over to another one, piece of cake, no issues, no worries. And so that has been a very important thing to do. Now, the next one, I thought I'd very briefly talk about achieving quality. This is about as far as we're going to get from mass specs. So to those of you who are less interested in quality, don't fear. But like I said, without all this quality, without these quality controls, etc., you don't know that the data that you're generating from your mass spec is actually as valid as it could be. Of course, validity and accuracy is a spectrum. You can sit anywhere on that. Depends on what the purpose is. We like to be at the very accurate, very reliable, no errors in the thing. And that means that there is a lot of quality related things that we have to do. So we have to get our lab accredited, we have to validate all our methods to particular international standards, we have to get those methods accredited, we have to make sure the machines are always working exactly properly, and we have to run all those quality controls every single time we do samples. So if someone wants to run one sample of something, you still have to do 10 quality controls. If someone wants to run 100 samples of it, you still do 10 quality controls. You have to do them every single time. So let's go back to the actual purpose of this, is to talk a little bit about mass spec. So there are 4 kind of classes, I suppose, of mass specs that we really use for different things. First one is GCMS. We'll all know about GCMS. GCMS take samples with small volatile molecules and then volatilize them, pass them through a column with some carrier gas, detect them. So we have some GCMSs, we have some GCMSs, and we have GCs with other detectors on them like FIDs and ECDs and so on and so forth. And we're going to talk a little bit more in detail about what we do with these later on, but for now, this is just high level. The other type we have is LCMS systems. So we've got a lot of triple quads, we've got some high-res systems, and we have some single quad MSs as well. And every single one is interfaced to an LC system. There is no mass spec which is standalone that we're infusing random samples into to check our sort of synthetic products are correct or anything like that. It's all coupled to an LC. Every single method has chromatography up front. The fourth type of MS that we have is isotope ratio mass spec. Now, isotope ratio mass spec we use for one or two particular applications, as the name tells you, to look for the ratio of particular isotopes in products. Again, we'll talk about what those do and tell us a bit later. And the fourth one, the unloved cousin, is ICP-MS. So elemental analysis of various different things, heavy metals, alkalis, etc., in a range of different matrices. So we'll quickly whip over some of these other types of MS systems. My specialty is LCMS, so I'll only very briefly touch on some of these different types. GCMS, we have about half a dozen or so of them. They are used pretty much exclusively for analysis of environmental samples. So that is things like wastewaters, it is things like soils, it's things like industrial wastes, it is sites that companies might be trying to remediate because they want to build on them, or perhaps someone has discovered that there's drums of oil buried in their backyard in their cheap house they bought. Someone might send us some samples and test them for any of these different things. So polyaromatic hydrocarbons and different volatile compounds, PCBs, halomethanes. And of course, there's a couple of non-environmental related applications in there as well. So things that smell, of course, are primed to be analysed by GCMS. So terpenoids, for example, and hemp is something the hemp industry is keenly interested in, and knowing the different amounts and ratios of different things that give them their different organoleptic properties, and taints in dairy products as well. So generally, dairy companies do an exceptional job of standardizing milk, so it tastes pretty much exactly the same, regardless of what bottle you pick up. But there is a number of things that farmers do, which has an impact on what cows are eating, and then consequently on what metabolites or what small molecules come through in their milk, and we refer to those as taints. So some of that we do by GCMS as well. And you'll see now I'm being deliberately vague about some of these things. This comes back to what we were saying before about, unfortunately, we can't tell you too many details about some of these things, but hey, it is what it is. Isotope ratio mass spec. There's a particular application that we do a lot of, and we have, again, about half a dozen or so isotope ratio MS systems. And what we do mostly with them is the analysis of what's called C4 sugars in Manuka honey and other honey types, in fact. So depending on certain types of plants, they can biosynthesize sugars with one of two or arguably three different synthetic pathways. And that results in there being slightly different ratios of carbon-12 and carbon-13 in the sucrose that they produce. Now, there is a second issue where the honey that we eat, that we find in jars on the supermarket shelf, was in fact meant to be the food for bees over winter. So when beekeepers take honey out of their hive, they're actually taking away the food that the bees have laboriously collected. And so they get fed sugar syrup during these hard times. Sometimes some beekeepers accidentally feed bees a bit much sugar syrup, and sometimes that sugar syrup ends up in the honey. And that's what we can detect with isotope ratio mass spec. So we take a sample of honey, precipitate the protein with sulfuric acid and a tungsten salt. We collect the protein, we clean it, we dry it, we weigh it, and we combust it in this isotope ratio system to produce CO2 and nitrogen oxides, and then we measure the abundance of the for example, the carbon 12 versus carbon 13. And based on that, we can infer indirectly whether or not a particular sample of honey has been adulterated accidentally or otherwise with sugar syrup. And that's quite a problem for the industry because some people do it a little bit more than they should, and some people do it on purpose. And sometimes when that honey goes overseas, they get caught out, and you get news articles cropping up like this that says X many percentage of honey jars from certain countries are adulterated. This is very damaging to our reputation. ICP-MS. So again, I'll only very briefly touch on this because, to be honest, I'm not involved in it that intricately. So we have only 2 ICP-MS systems, but they run pretty much 24/7 doing elemental analysis on different sample types. So again, for environmental samples, there are things like heavy metals that you would want to detect in certain effluent streams or soils. And in things like dairy products as well, there are elements that might be present, which either should or shouldn't be, depending on what the animals have been fed or what has been put on the pasture or what's in their drinking water, etc. So what I thought I would do now is just briefly go through a couple of examples of some of the stuff that we do with LCMS in particular for honey and for some of the other products as well. Because like I said, LCMS is my area of expertise. So we've got about a dozen or so different LCMS systems, and like I said, they're all fairly similar. And what that means is that we can set up particular tests on them and move them around to achieve that throughput that we need. Now, there are 3 sort of key tests that we do for honey, for example. So there's some of these tests are basically handed to us to do on a platter or in legislation, perhaps you might say. And some of them you have to create opportunities yourself. But these 3 are examples of ones that are legislated. So first one is a set of chemical markers, for example, that we analyse in honey. So a few years ago, the Ministry of Primary Industries decided that it needed to do something about all the adulteration of honey that was happening in the market. And they did a science program with some untargeted analysis and some targeted analysis of different honey types, and they came up with a suite of chemical markers that could be used to tell whether or not a sample of Manuka honey was authentic. And consequently, they wrote that into some of our national legislation that said anyone who was exporting honey had to test their honey for these particular things. And so that, for example, is one of the things that we might do with LCMS. 2 of the other things, which are perhaps slightly more topical, are residues that are in honey. So one that most people haven't heard of is one called tutin. So tutin is a metabolite that is found in the tutu bush, which is native to New Zealand. And when you have certain insects that feed on it, they excrete the secretions from this plant, and it contains this metabolite. And then the bees collect it, they put it in honey, and then when people eat that honey, they get poisoned and get very sick. So for a long time, this was quite a mystery. So when Captain Cook et al. Came over the very first time, they would release livestock into certain areas, and some of it would die because it was munching on these tutu bushes that were around. And the same problem still exists today, or in a lesser form, where bees collect it, put it in honey, and it makes people sick. So we have a test where you measure tutin in honey, and here, for example, is just an illustrative chromatogram showing what that might look like. So that's 100 parts per trillion of tutin in honey. Another one which is quite topical is glyphosate in honey. So glyphosate has been a big issue in the media for the last year or 2, especially with what's been happening overseas, with people suing Monsanto and the likes for getting cancer from being exposed to glyphosate. But as a residue, there is a lot of legislation around it. Again, if you want to export products to certain markets, then there are requirements that you have to meet. So we have a particular glyphosate test where we take the honey, we dilute it, we derivatize it, we run it on an LCMS system, and you measure it down to a very, very low level indeed to make sure that it is compliant with the legislation. So that sometimes annoys beekeepers because we're detecting it down to what they describe as sort of 1/10 of bugger all, and no one should care about it. But of course, there are rules for reasons, and there's not much that we can do about it. So say, don't shoot the messenger. Now, in the dairy space, for example, there's a handful of different things. Well, there's several handfuls of different things, in fact, that we might like to measure. So there are detergents in milk. So When milk is collected from farms, it goes in tankers. They have to clean the tankers out periodically, and sometimes they use detergents for that. And of course, you don't want detergents being residual inside the tankers and the lines, because it ends up in the milk supply. And if someone finds it, then dairy companies get in trouble. So we do things like that. There are residues like 1080 and chlorate in milk. There are other sterilizing agents like chlorhexidine. One of the more prominent ones has been the issue with melamine. So several years ago, it was found that there were some companies who were adulterating milk powders and infant formulas with melamine, which is a very small nitrogen-rich molecule, which if you spike into dairy products, it artificially increases the nitrogen level, which affects the price and payout that people get for it. So as a consequence, we developed a test to look for melamine in dairy products. Again, going down to about 1/10 of bugger all. And here is a chromatogram that illustrates that. Now, there's a couple of other different bits and pieces that we look at and measure as well. So like I said, we're kind of in the drug space to some extent. So if you are a renter, then it may be common that your landlord will periodically come around or send someone around to swab your walls with a little swab containing isopropanol. And what they're doing is they're extracting from the surface of your kitchen, bedroom, bathroom, garage, etc., any potential residues of methamphetamine that might be there. So we take those swabs, we digest them, we extract out the methamphetamine, and we run it by LC and SMS. And again, we measure it down to very, very low levels to make sure that people are compliant with whatever those legislations are. Of course, it's always a really fun game to look through the methamphetamine results and look for the high ones, and then try and guess, if you go to Google Maps, what the place will look like. Although you will be surprised, because as people say, it's a very pervasive drug. It penetrates all layers of the socioeconomic hierarchy. So there's all manner of places you find it. Like I said, we do a bit in the hemp space. Down the bottom there, for example, is an example of some THC in a hemp product at a particularly low level. What you may have noticed from many of these chromatograms as well is that the retention times are all quite small. So most of them, in fact, are less than a minute. What that means, of course, is that most of the methods are exceptionally short as well. So in our lab, there is pretty much no method that's longer than 10 or 15 minutes in total length. Columns longer than 10 centimeters are outlawed, basically. If you get a smack on the wrist for buying a 15 or 25 centimeter column. Don't think about doing like a 4.6 millimeter internal diameter, like that's not allowed. So all these methods are exceptionally short. They're designed, they're totally free of interferences, as accurate as they can possibly be, and have a very, very rapid turnaround as well. Now, what I thought I'd do next, because these have all been quite high-level examples, and I appreciate that's not necessarily what you guys are interested in. I mean, I like to know the details of these things as much as the next man or woman who's interested in an aspect, but again, a lot of them we just can't for reasons I've mentioned earlier. But here's two examples, three examples, in fact, where I thought I would elaborate just a little bit more because they're kind of prominent within our food area. So, case study number one that I've got here is the case of A1-free dairy products. So anyone who has been to the supermarket and paid attention to the milk section, or who has children and has bought infant formula and has paid attention to the infant formula section, will note that there is an increasing number of these A1-free or A2 type dairy products that are appearing in the market. And what that means is just that they are free of a particular isomorph of beta-casein, which potentially liberates some peptides when you digest them that make your guts do unhappy things. But the problem is that because this is an emergent market, the testing for it is very emergent as well. There's no standards, there are very few labs who can do it. So of course, this is where we come in. So here's some actual science for you guys, very briefly. One of the first approaches that we tried to use to authenticate these type of products was an intact protein analysis. So we would take the dairy product, we would extract out of it the intact protein molecule or moiety or whatever you want to call it. We would purify it slightly, and then we would run it on a reverse phase gradient using a protein type column, and detect it with either UV or high-res MS. And so when you do that with something like fresh milk, it works pretty well. There are some limitations. It's not particularly sensitive. Not everything is really baseline resolved. I looked at their spectra first. I was like, oh, what is that? It's not like a nice peak with a flat baseline, but it's actually pretty good. So here's an example of what you get there. You've got 3 peaks. You've got an A2 beta casein, an A1 beta casein, and another interference. But they're all fairly well resolved from one another. And when you run them on the MS, you get really beautiful spectra as well. Lovely envelope corresponding to the different charge states from about 15 plus up to about 30 plus up this left-hand side. And what we do is we can then take that data, pass it through a deconvolution package to calculate the high-res parent mass, if you will, and check that against what's in the literature and say that, yes, this in fact matches. We touched on mass accuracy before. Usually we can calculate these masses and they match within about 10 parts per million, which is pretty good for deconvolution of all this garbage here. Now, like I said, that works great for milk, but when you come to something like infant formula, which is a little bit more complex, you don't end up with something that's quite as beautiful. You end up with a big broad peak and the spectra a little bit more complex as well. And so when we first did that, we kind of said, ooh, not quite sure what to do with this. It doesn't quite work as well as the fresh milk. So that's when, of course, you move to doing a digestion of some type. You take the protein, you extract it, you expose it to a protease of various types. Of course, there is more than one. And so we've done a lot of this research as a foundation to try and build targeted methods for analysis of dairy products. So we take the proteins, we digest them, we run them on high-res MS with data-dependent MS2. We map those MS2 spectra against the proteome of the cow, and then we find which peaks correspond to which position within the protein, and then we pick the peaks which correspond to the substitutions that distinguish one variant from another. And from that, we can then build a very short, very fast, targeted method that we can use to do 1,000 samples a day, for example. Here's an example of that here. So you go from the mishmash of peaks in the previous one to something like this, which is relatively clean, relatively simple, relatively targeted. So number 2, case study number 2, is the old Manuka honey story again. So we touched on it a little bit before, but again, there's some background. So Manuka honey is a honey which is arguably exclusively native to New Zealand, produced from the nectar of Leptospermum scoparium. Now, Manuka honey is quite expensive. Again, you go to the supermarket, you look at the jars you've got there, they might be anywhere from sort of $10 to $40 or $60 perhaps. And you look at that and say, oh shit, $60 is a bit much for something I'm going to put on toast. But of course, we don't get most of the really expensive high-grade honey at the supermarket. It goes overseas, it goes in duty-free shops, it goes into totally different markets. And there are some eye-watering prices that come up there as well. So a jar no bigger than this cup, you can sell for $2,000 or $3,000 to certain people. Again, not really toast honey. It's given as a sort of a gift, you know, as a substitute, for example, as for a bottle of single malt whiskey or something like that. Not the same thing, of course. We don't recommend single malt for breakfast, if you can with toast. So what we did around the space is we started doing some work for the UMF Honey Association in about 2013. The UMF Honey Association are an industry body that maintains some quality standards relating to Manuka honey. And the first thing that they did is they contracted us basically to do some R&D for them and run some samples by LC HRMS. And so the first thing we did is we analysed some nectar samples, because of course honey is produced from nectar, and if you know what's going on in the nectar, then in theory you should be able to figure out what's happening in the honey. So we collect some sorry, I shouldn't say we did nothing. We sat on our asses in the lab, and answered emails. Someone else went out in the field, pulled flowers down with a pipette and sucked out 5 microliters and put it into a tube. So I can't take credit for that aspect of it. But we did, however, run them through the MS, and these are the kind of results you get. So between nectars of different floral types, you can pretty clearly see with your eyes alone, you don't need any bioinformatics for it, that there are some quite stark differences in the composition of different nectars. Again, not totally surprising. They are from different species. They're going to have different metabolomes. There'll be totally different markers that will be present in one to another. So we said, great, we've got separation between different nectar types. In theory, that should translate into honey. Here's some examples of what it looks like in nectar. So again, we look for particular markers, do some PCA, get decent separation between certain things, find markers that appear to be significantly upregulated in one nectar type compared to another. And so that looks like a good one. We'll have a look for that in honey. So again, then we move to honey. So here are some chromatograms, for example, of the same 3 honey types, but taking whole honey that the beekeepers put their hand on their heart and say, this is the most pure manuka or most pure kanuka that you'll ever find. And we say, okay, yep, sure, we believe you. Run it through the mass spec. This is what you get. So again, some very different chromatograms between the different floral types. Because we have high-res MS, we can look into the data. We can see what ions are there. We can calculate what the monoisotopic neutral mass is. Calculate the formula. We can search the literature, see if there's anything there. If there's something in the literature that matches, we buy the standard piece of cake, in it comes. Run it on the mass spec. Matches, fragmentation patterns, same. There's your answer. Marker identified. So there's three that have come up variously as being useful for this. So this big guy here in the middle is a molecule called 3-phenylactic acid, which is present in kanuka and manuka honeys. There's this little guy down here, which is still present at thousands of milligrams per kilo, called leptospirin, which is a unique marker for manuka honey. And there's this little guy down here called lepteridine, which was discovered at this very own university, in fact. So we've said, yes, here's your data, here's your report, these are good markers. Let's turn it into a targeted test. So that's what we do. We take that data, turn it from a 30-minute untargeted metabolomics thing into a 3-minute targeted HPLC fluorescence type assay. And there's an example of it there. And that is honey. So I think this blue one is a standard. This guy here, which you can just see, is a good manuka honey, and down here there's a not so good manuka honey. Third and final example that I just wanted to briefly touch on is PFAS. So PFAS is a fairly topical class of pollutants called perfluorinated alkyl substances, which people are finding spread all throughout the world because they're used in repellent coatings and Teflon and your grandma's socks and all sorts of things. So they are ubiquitously spread throughout all sorts of products. They are persistent, they are bioaccumulative, and they are potentially toxic. And are characterized by this perfluorinated chain and this polar group. So they're kind of surfactant-like. So we had a client come to us and say, well, we're remediating the site. We want to kind of follow the remediation process, and we want a little bit more insight into what's going on than just what your targeted test does. So we got all their samples after they've been through various rounds of remediation, different processes, and we ran them on our targeted LC-MS method, which was 10 minutes long. And then we ran them on our untargeted high-res MS method. And you can see here, for example, is some chromatograms indicating the changes as you go along. So top one, very polluted, middle one, halfway through the remediation, green one down the bottom there, very near the end of remediation. So this corroborated what we were seeing in the targeted LC-MS data, and also there were some other bits and pieces in there that we found that we wouldn't have otherwise seen with that targeted approach. So it's supplementary and confirmatory, or supporting, I suppose you could say. And that's really what we do. It's what we do with mass spec in our commercial environment, at least. We develop targeted tests reliable, high throughput, provide them as an off-the-shelf product for people to use. And if they want more information than what that provides them, we can do bespoke R&D type exercises to support that. And that's mass spec in our commercial environment. Thank you for listening.
Do we have any questions? We have some time for questions. Yes.
Great talk. I just wanted to know, are there actual regulations now on amounts of methamphetamine in the swabs?
Yeah, there is. So there was a regulation that was quite high and arguably a little bit hastily put in place. And then some number of years later, there was a report released from the government, the Gluck-In report, they call it, where they reduced that limit by, I think it was tenfold, in fact. And so there was a lot of, they increased it by tenfold. So there was a lot of very angry people who had spent hundreds of thousands of dollars remediating properties that the government then turned around and said, oh, actually, those limits are fine. We're raising it to, yeah, this is the safe level now, down here. So yes, there are some in place.
Any other questions?
Hi, thanks for the great talk. So I think everybody could be a potential customer for the A1-free formula product. So
sure.
My interest will be what's the actual criteria to define product as being A1-free?
It's a very good question. There is none. There is. It's one of those. It's one of those problems that because the area is really freshly emerging, and in fact, there's no agreement even on the test methods yet, let alone what a potential residue limit should be for any A1 beta casein that's present. So generally, the industry uses a limit of what's called 1% relative to the total amount of beta casein. So of all the beta casein that's in the sample, less than 1% of it should be of the A1 type. But that's totally voluntary. It varies from company to company and country to country as well. So it's a difficult area.
Do we know how much of the A1 is actually being given the peptide that we don't want? Maybe that would be worth it. Afterwards, we could figure out.
Yep. So we broadly speaking, as in science, knows a little bit about it. The issue is that the efficiency of that digestion and the production of those peptides varies tremendously depending on what the conditions are. So you can try and do animal studies where you extract the contents of their gut or their plasma or their other bits and pieces, and then assay it for that particular peptide, which we have done as a supporting thing for this project. But it doesn't necessarily tell you about what sort of yield you're getting or where it's going or what have you. You can do simulated digestions as well, but they are wildly variable in what we do because, of course, there's a lot of different proteases and things, a lot of different molecules that are contributing and changing the nature of their digestion in a person or an animal compared to when you're doing it in the lab. And we've got 3 enzymes from the same order actually doping it. So, yeah, there's some information.
Do we have?
Man in the back?
Oh, yeah, I was just curious. 24 hours seems like insanely quick turnaround for analytical labs.
Yep.
Well, like QA review and everything.
Yep, it absolutely does. So what happens is we'll do a quick run through, for example. So 9 a.m. yesterday morning, for example, a courier turns up, drops off a package with a sample of honey in it. So that sample goes immediately over to our sample reception team. And what they are going to do is they're going to open the package, inspect it, look at the submission form, register it into a central database of samples, apply a unique identifier to it that traces it through the lab. And then probably an hour later, it goes into another part of the lab. So what they will do is they'll homogenize the sample, they'll weigh it out, and they'll add some extractant to it. And again, doesn't take more than an hour. In theory, it could take 5 minutes. That tube then gets collected up with a bunch of other tubes, other people's samples, which might take a couple of hours potentially. They all get moved then to a robot that does some liquid handling. and that robot will take aliquots of it, do the derivatizations, do the dilutions, etc. And that might take an hour, let's say. So once you've then got all those samples moved into a plate or vials or whatever it may be, they can then go on an instrument that afternoon. So they'll run overnight because the methods might only be 3 minutes long. Let's say you've got 100 samples to do, that's 300 minutes. So you can run several lots of a couple of hundred samples overnight. The next morning, first thing, some technicians come in, 7, 8 a.m. They flick through the data, look at all the peaks, make sure the integration is correct, paste the data into some of the checks or quality control metrics against predefined standards, pass that to another person who then does another check on it to make sure that they've done their job correctly. And that can all happen by 9 or 10 that next morning. The data goes back into the centralized data handling system, and automatically the reports go emailed out straight to the clients. And that's how it happens in 24 hours.
Pretty impressive, and not a whole lot of room for error.
No, there isn't. So if something does go wrong, then we do have to fess up and bring people up and say, yeah, actually there is a problem. Dickhead over here put the plate in the robot backwards, or someone forgot to fill up the robot with extractant solution and all the wells were dry, or it'll be another 24 hours, sorry. Usually people are fine with that, as long as you're honest and upfront about it. But sometimes, of course, there will be people who are relying on that turnaround. So it usually works out, but not quite always. There is, of course, room for mistakes. We do have people working on these things at some point. People
and
do make
instruments can break down in the middle of the night.
They do, yes, they do that.
They do have sick days.
They do have sick days, yep. Hopefully not more than a day or two.
Yeah.
Like I said, we try and get them repaired pretty much ASAP, so they're back on their feet. They've got work to do and money to make. We'll be working tomorrow. So, yeah.
Do we have any other final?
I have one more. And for method development, how much time you allowed to develop a new method? You have that in the system, or you said?
So it depends. It depends how much information you get upfront. Really, I mean, if someone comes to you with a incredibly vague problem like develop a method to authenticate Manuka honey, that's not like a 2-week development process. That's like a multi-year thing. Of course, if someone comes to you and says, can you measure this particular compound in this particular matrix, and you can go to the literature and see that someone's done half the work for you already, then you can do it within a month.
One final question, we have to move on. Yeah.
What type of internal standard do you use for your matrix effect?
We use for some methods we use isotopically labeled internal standards, whether that's deuterium labeled, carbon labeled, nitrogen labeled, and for some methods we do not have internal standards, but we rely on other approaches to check or correct for matrix effects.
I have the most important question. Are you hiring?
We're always hiring. We're always hiring. Absolutely.
All jokes aside, if you are hiring, please do let us know when we advertise through MASH as well.
Sure.
So we appreciate that. We have to move on, but if someone wants to catch up with you, they